Welcome back. What we're going to do in this video is talk about hemoglobin. And what we're going to find with hemoglobin is that it's significantly more complex than myoglobin. Most people, when they think of hemoglobin, they just think of oxygen binding. But as we'll find, there are many functions associated with hemoglobin. Some of them are also associated with other enzymes of the red blood cell, but we'll find that the function is incredibly complex. So let's just review briefly what we talked about in the last video. So we have our porphyrin ring, right? And I'm drawing it in the flat mode, right? So this is if you were sort of looking at it from the side view. And recall that they just had your porphyrin ring over here, right? And then you also have your histidine residue that chelates from the bottom. And then you also have your oxygen molecule that is going to interact with the top of the porphyrin ring, right? So there's your oxygen. And just recall that just like in the case of myoglobin, it's going to come in at an angle theta, and theta is about 120 degrees, okay? It's going to vary just a little bit depending on the location of the lone pair electrons, but generally it's going to be about 120 degrees, okay? Now, in terms of the setup of hemoglobin, hemoglobin exists not as a monomer, but as a tetramer, okay? So hemoglobin is going to have a structure that looks like this. So you have these 4 globin chains that are going to interact with each other, okay? And so this is your setup for hemoglobin. And what's important to understand about this is each one of these contains a heme, okay? So now we can come back and look at myoglobin and ask the question, per myoglobin, how many oxygens can be bound? Well, it's just one, right? Because there's one, it's a monomer, and each monomer has one heme. So you can bind one oxygen molecule per myoglobin because myoglobin has one heme, right? So the question now becomes, per hemoglobin, how many oxygens can be bound? And the answer is 4, right? Because per hemoglobin, you have 4 hemes because there's 4 globin chains, right? So what you can imagine is that, well, just suffice it to say for now, we'll go into more detail later, but hemoglobin can bind 4 oxygen molecules, okay? And it's going to do so in this orientation right here, okay? Now, before we really even get into the oxygen binding, there are some other things we have to understand about the red blood cell, okay? So for that, I'm going to come down here, okay? And I'm just going to draw a red blood cell, okay? So here's your red blood cell, okay? And in general in this video, this is going to be a little bit of a longer video, but we're going to talk about all the functions of the red blood cell. Now, what I want to do is I want to show you a reaction, and there's an enzyme that's associated with the red blood cell, okay? And the enzyme is called carbonic anhydrase. And in future videos, we may look even at the mechanism of carbonic anhydrase, okay? And so effectively what this enzyme does is it interconverts something called carbonic acid. Converts carbonic acid with CO2 and water, okay? Now, you may look at this reaction, you say, oh, well, it's 2 equilibriums. How can we treat it with Le Chatelier's principle? Now, what you have to first realize is that the actual reaction that carbonic anhydrase is catalyzing is this one right here, okay? This is the reaction that carbonic anhydrase is catalyzing. Now, it's reversible, but what you have to understand is that carbonic acid, which is denoted here, this is carbonic acid, it's very unstable, okay? In fact, there's some individuals who believe it might not even exist at all like that, but it's not going to last very long. And so if you're running in the leftwards direction from CO2 and water, carbonic acid is going to rapidly fall apart into bicarbonate and protons. Okay, and of course the protons are going to exist in the hydrated form as H3O+. Okay, this right here, this is bicarbonate. This is bicarbonate. Okay, and So effectively what this enzyme carbonic anhydrase is doing is it's interconverting CO2 and water with protons and bicarbonate. It's just that the initial product that's released is carbonic acid, but it rapidly falls apart. Okay, so let's think about respiration. Okay, because in large part some of the binding of oxygen has to do with respiration. So let's say I exhale. Okay, if I'm exhaling, what am I exhaling? Well, first of all, I'm exhaling water. In fact, the reason you see, if you breathe on a mirror, the reason you see the condensation on there is because that's water that's produced by metabolism. As we'll find when we get to metabolic pathways, water is one of the waste products. But one of the other waste products is carbon dioxide, which we've shown next to water in this reaction. Okay, so both water and carbon dioxide are waste products. Okay. Now you have to think about what am I going to produce when I have actively metabolizing cells. Okay, and you'll see that there's a method to this madness. What I'm doing. Okay, if I've got an actively metabolizing cell, so let's come down here. Let's say I have a cell. Okay. Let's say I have a cell that's actively metabolizing. What am I going to be producing? Okay. Well, we just mentioned we're going to be producing carbon dioxide, right? That's one thing. And so what we'll find is that the tricarboxylic acid cycle, or you might have heard of it as the Krebs cycle, that produces 2 carbon dioxides. There are certainly many other reactions that produce CO2. Okay. Another thing that's produced by actively metabolizing cells is H+. And of course, that's probably going to exist as H3O+, as the hydronium ion. But what you would find is that if you took the pH directly outside actively metabolizing cells, the pH is going to be lower there than it is going to be, say, in a cell that's static. Okay, so if you have an actively metabolizing cell, you're going to be producing hydronium. Okay. And another molecule that you're going to find, and it's debatable as to whether or not it's produced by the actual cell or not, but it's going to be associated. The actively metabolizing cell is going to be associated with a molecule called 2,3-BPG, and 2,3-BPG stands for 2,3-bisphosphoglycerate. We're going to come back to that a lot later, but suffice it to say that when a cell's metabolizing, it's going to be producing a lot of that. Okay. Now, for now, what we're going to worry about is hydronium and carbon dioxide. Okay. And these are 2 things that are produced by actively metabolizing cells. Now, you can imagine that if I'm loading, so CO2 and hydronium, they're going to leave the actively metabolizing cell and end up in the blood, right? I hope that makes sense. So CO2 is going to leave the cell. Acid is going to leave the cell in the form of H3O+. So you can imagine that outside the cell in the blood, the concentration of H3O+ and CO2 are going to rise, right? Well, it turns out that both these things are not exactly the greatest thing for a cell, right? So if I have an actively metabolizing cell, there's going to be a lot of H+ to begin with. But if you think about it, if I'm releasing CO2 into the blood by Le Chatelier's principle, right? If I raise the concentration of this product, right, what happens? I initially shift the equilibrium of carbonic anhydrase towards carbonic acid, but then that rapidly builds up. So this will build up, right? And then it'll fall apart into bicarbonate and hydronium ions, right? And you have to realize that, okay, bicarbonate's a base, yeah, but you can sort of view hydronium as a strong acid, okay? So the strength of hydronium as an acid far outweighs the strength of bicarbonate as a base. So the net effect is a lowering of the pH. Now, if we come back up here to the red blood cell, what you have to imagine is you have your hemoglobin molecules, right? So your hemoglobin, right, was just this tetramer of globin chains, right? And attached to these globin chains, you have histidine residues, right? So you have histidine, okay? So you have your histidine residues, right? And what you have to imagine with the histidine is that, okay, I have this imidazole nitrogen, right? And you also have to realize that the pKa of that histidine nitrogen, specifically this one, is about 6, Okay. Now, it is true that the pH of the blood is about 7.4, and yeah, you could make the argument that 6 is not really in the buffering zone of histidine, right? The blood is not with the pH of the blood is not within the pKa plus or minus 1 pH unit, right? But as you start to lower the pH of the blood, because you're building up these hydronium ions, okay, you start to increase the propensity of these imidazole nitrogens to bind protons, right? So let's say I'm going into some sort of acidosis, right? It's just an equilibrium, but let's say I'm going into some sort of acidosis, right? So I have my hydronium ions, right? And so what's going to happen is there's going to be a lone pair on this imidazole, right? And effectively, this is going to be a proton transfer, right? So nucleophilic attack, loss of leaving group, right? So what happens is you end up protonating the imidazole nitrogen. And so what the hemoglobin, some of the imidazoles on there serve as is a proton reservoir, okay? So when you have low pH in the blood, hemoglobin actually serves as something that can pick up extra protons and bind them, which effectively raises the pH back to normal levels, right? And so that way, whenever you have, okay, so let me come over here and do this. Whenever you have, let's say you have too high of a pH, right? Let's say your pH is a little bit, let's say your pH is, oops, that shouldn't be that way. Let's say, if I can draw this correctly, let's say that your pH is a little bit too high, right? So maybe you have this in the protonated state. Let's say your pH is a little bit too high. Well, it turns out that you can do the reverse reaction. So instead, this can come in here and deprotonate the imidazole, right? And this should have had a positive charge, right? So it turns out that you can, when the pH rises to a significant extent, you can actually deprotonate the imidazole and lower the pH back to normal levels. So there's a careful equilibrium between the protonated state of the imidazoles on the hemoglobin. There's an equilibrium between the protonated state and the deprotonated state. And that equilibrium allows hemoglobin to act as a buffer. And specifically, it's the histidine residues that act as the buffer, right? And by this reaction right here, if you load the body up with carbon dioxide, what happens? Well, carbonic anhydrase favors the production of carbonic acid, which rapidly falls apart into protons and bicarbonate, okay? So if you're producing a lot of CO2 through metabolism, you're going to produce a lot of protons, and the hemoglobin histidine serve to pick that up. Okay. Now, it turns out that carbon dioxide is not soluble in the blood. Okay. And to understand that, we need to actually analyze the structure of CO2. Okay. So if I draw a molecule of carbon dioxide, right? Now, you might be tempted to say it has a dipole moment on it, but remember that these two dipole moments that point towards the oxygens cancel each other out. So carbon dioxide itself is nonpolar, okay? So it's not soluble in the blood, right? So it would make sense if we had a way to transport carbon dioxide in the blood, and it turns out that red blood cells are perfectly capable of doing this, using carbonic anhydrase, but also hemoglobin has a function in it as well. Now, we've already, whether you realize it or not, we've already talked about one of the ways CO2 is transported. It's in the form of bicarbonate. So if you look at bicarbonate, you can see that it has a net dipole, right? There is a net dipole in bicarbonate. In fact, there's one that goes towards the oxygen from the proton, right? So overall, bicarbonate is very polar, and therefore it's soluble in water, which in the blood is water mostly. So therefore, bicarbonate is soluble in the blood. So what's the purpose of doing this reaction? Number one, it's solubilizing CO2, right? In the form of bicarbonate. Now, in the process, we produce protons, but that's okay, right? Because the red blood cell has hemoglobin, and histidine residues can pick up the protons and act as a buffer. Okay, so that's all good. Okay, now there's another form of carbon dioxide that we can have, and it's as a carbamate. What's a carbamate? Well, essentially what you're going to have is you're going to have the protein chain, right? And let's say you have a lysine. So 1, 2, 3, 4. 1, 2, 3. Yeah, so you have a lysine, right? And I've specifically drawn it in the deprotonated state because what you're going to find is that the lysine can act as a nucleophile. Okay, it can do a nucleophilic attack on this carbon, and that kicks this pi bond out here. So what do we generate? Well, we generate another equilibrium state, and specifically if we load the body up with CO2, what do we get? 1, 2, 3, 4. Well, we're going to get something that looks like this. Right? So we get this molecule right here. This is the carbamate version of lysine. And it turns out that this lysine residue is attached to hemoglobin. So not only can hemoglobin serve as a buffer for the protons that we produce from carbonic anhydrase, right? But it can also serve as a binding point for carbon dioxide. So it turns out that when carbon dioxide is elevated in the blood, right? Carbon dioxide is elevated in the blood, it will react with carbonic anhydrase, but also some of it can be attacked by the lysine residues of hemoglobin, okay? And this particular reaction is non-enzymatic, and it's actually reversible. So now that we've talked about what happens during high CO2 levels, let's talk about what might happen if we have low CO2 levels. So let's say we're in an area of the blood where there's low CO2. So now we're simply, again, applying Le Chatelier's principle. And if you remember from the last video, I said that red blood cell function is really just an exercise in Le Chatelier's principle, right? So let's say that I lower PCO2, right? So the partial pressure of CO2 gets lowered. What's going to happen? Well, you're going to shift this whole reaction to favor the production of the products, at least in the way that I've written it. So what's going to happen? Well, what's going to happen is the protons are going to combine with bicarbonate to form carbonic acid, and then carbonic acid is going to react with carbonic anhydrase and release CO2 and H2O, and that's, of course, going to raise the concentration of CO2 back to normal levels, right? Now, if you imagine, if you're combining protons with bicarbonate, what happens to the proton concentration? It's going to drop, right? If you're combining protons with bicarbonate to form CO2 and water, proton concentration drops, right? So if we come back up here to the red blood cell, if proton concentration is dropping, which one of these equilibriums is going to be favored? It's going to be this direction, right? Because if proton concentration is dropping, what's happening? You're having to get the protons from somewhere, right? So water is going to pick up the protons from the imidazole, and you're going to form hydronium, right? So that's literally where the protons come from in this reaction. You could really think of them as, what is it, H3O+, right? Now, in that case, this equation's not balanced, but effectively the protons are hydrated, right? So if for some reason CO2 drops, we're going to consume the protons with bicarbonate to form CO2. But if we're consuming protons, they have to come from somewhere, right? So one of the sources they come from are these protonated histidine residues. So water will come up, nucleophilic attack, abstract the proton, we'll generate hydronium, and then we'll do the carbonic anhydrase reaction, okay? Likewise, what happens if CO2 is high? We're going to produce hydronium, right? So I'll go back to this dark, this color, right? If CO2 is high, which direction do we run? And we run in the direction of proton production, right? So if protons are being produced, what's happening? Right, we're going this direction, right? So if we have lots of hydronium, the imidazole nitrogen is going to attack the proton, loss of water, and we end up generating protonated histidine. So the direction of this carbonic anhydrase mechanism is really dependent on two main things, carbon dioxide concentration and proton concentration. But really what we're mostly concerned with is the carbon dioxide concentration. And one of the sources of protons is this reaction with the histidine. Okay, just a simple protonation, deprotonation. Okay, now if we have high CO2, right? If we have high CO2, which one of these reactions is going to be favored? Which direction? If we have high CO2, we're going to run it in this direction, right? So if high CO2 is that if CO2 is elevated in the blood, we're going to run it towards the direction of the carbamate production, right? What happens if CO2 gets lowered in the blood? What happens? If CO2 gets lowered in the blood, we're going to reverse this. Right? If CO2 gets lowered, we're going to reverse the carbamate production and regenerate the lysine residue and the carbon dioxide. How does that happen? What's the mechanism? This lone pair kicks in here, and this bond comes out and abstracts a proton, right? Hope that makes sense. So if we have low CO2, carbon dioxide is going to come off of the lysine residue, and we're going to regenerate lysine, right? So when you think about red blood cell function, it's really critical to think about Le Chatelier's principle. And in general, what we've talked about so far is hydronium reactions, which is with the imidazole and carbonic anhydrase, and CO2 reactions, which is with carbonic anhydrase and hemoglobin lysine residues. Okay, so this is generally what we've seen so far. And we've seen several functions of hemoglobin up to this point. Now, another function of hemoglobin that we've sort of alluded to so far is protection from carbon monoxide. So if I have my iron, right, and I'm looking at hemoglobin, or excuse me, the heme from the side, right, recall that the iron, which I should put is normally in the 2+ state, right, can bind carbon monoxide, right? It can bind carbon monoxide. What's the function of that? Well, one thing that probably isn't apparent to you at this point, but hopefully it will be by the end of your probably Biochemistry 1, is that the respiratory chain, which is one of your electron transport chains that's responsible for ATP production, utilizes hemes, right? What happens if carbon monoxide gets into the respiratory chain? What happens to the activity of it? It shuts down, right? So if you are shutting down your respiratory chain, are you producing the 30 ATP? No. And now you're relying on glycolysis, which only produces 2. So if you're actively depending on glycolysis, is your cell going to live very long? No. So one of the ways carbon monoxide kills you is by shutting down your respiratory chain. Okay? And the specific enzyme it inhibits is called cytochrome C oxidase, and we'll get to that eventually. But that's a heme protein as well, binds oxygen. Oxygen is one of its substrates, and so if you get carbon monoxide into the active site, it shuts down. Okay? So one of the functions of hemoglobin is to bind carbon monoxide before it gets into your cell, right? Before it gets into the mitochondria, where the respiratory chain is, right? So if you can get carbon monoxide bound to hemoglobin before it gets in there, you're pretty much assuring that carbon monoxide can't get into the respiratory chain, right? And one thing they don't tell you usually in biology classes, they normally just tell you hemoglobin binds oxygen, right? But it turns out that approximately 1 to 2% of your hemoglobins are bound to carbon monoxide. That's sort of a significant number. So only 98 to 99% are actually bound to oxygen. And there are actually other molecules that can bind to the iron. Nitric oxide is one of them. Nitric oxide is a vasodilator. It turns out that nitric oxide can hitch a ride on the hemoglobin as well. But one very important function of hemoglobin is protection from carbon monoxide. So about 1 to 2% are complexed there on the heme iron. And so that just protects us from the effects of it. Okay? So that's one function. Okay? Kind of a short thing right there. Now, one thing that we also have to talk about is one of the enzymes in glycolysis. And this may seem sort of an odd thing to talk about, but one thing we'll talk about when we do glycolysis is there's an enzyme called phosphoglycerate mutase. So this enzyme is going to take phosphoglycerate, and specifically it's 3-phosphoglycerate. I'm just abbreviating the phosphorus, right? Or the phosphate, excuse me. And it's a reversible reaction. And phosphoglycerate mutase in the direction of glycolysis, the intermediate that it's going to go through is it's first going to phosphorylate. It's going to phosphorylate the 2 position, right? So, and then finally, the last part of the reaction is where it hydrolyzes off the phosphate at the 3 position, and you're left with what? you're left with 2-phosphoglycerate, right? And that's the reaction of phosphoglycerate mutase, okay? And so what we would do is we'd say that this is an intermediate, right? That's your intermediate, okay? Now, what is that molecule? Well, this molecule is called 2,3-bisphosphoglycerate, and we usually abbreviate it 2,3-BPG. Some texts and some things you might hear might just abbreviate it BPG, but to be absolutely specific, we'll call it 2,3-BPG, okay? Because there's also a 1,3-bisphosphoglycerate, but that's beside the point, okay? So 2,3-BPG gets elevated around cells that are actively metabolizing. Now, what does 2,3-BPG do? Well, 2,3-BPG, carbon dioxide, and H2O all favor the dissociation of oxygen from hemoglobin, okay? So now we're going to introduce to you a big concept. We'll come back to 2,3-BPG in a few minutes, okay? We have two states of hemoglobin. I'm just going to draw them as boxes, okay? Actually, let me do one as a box, one as a circle, okay? In this case, I'm going to designate this as the T state of hemoglobin, and this state as the R state, okay? So what is the T state? Well, the T state is, you can sort of think of it as like the tense or the tight state, okay? If you think of sort of someone who's uptight, they're kind of like, oh, I don't want anything new, you know? Get new things away from me. I'm so set in my ways, you know? They don't want anything added to their life. And the T state of hemoglobin is sort of the same way. In the T state, oxygen dissociates. So you can sort of view it in two ways. Number one, if you're in the T state already, it's not going to bind more oxygen. But if you have oxygen and you move to the T state, it's going to dissociate the oxygen, right? Likewise, if you're in the R state, that's sort of the relaxed state, kind of the hang loose state. You know, it's kind of like, you know, if I, you know, no, I'm willing to try something new. You know, come on, hang loose. Come on. You know, come on. You know, I want something new. I'm willing to try some new things. So oxygen will bind to hemoglobin in the R state. Likewise, if you are in the T state and you move to the R state, oxygen will bind. So you can sort of view the R state as having bound oxygen. Okay, this is the state that favors oxygen binding, and T state favors oxygen dissociation. Okay. Why is this important? Well, it turns out that if you remember our three waste products, okay, and 2,3-BPG technically isn't a waste product, but for the purposes here, we'll sort of consider it that way. We'll generally say these are things produced by actively metabolizing cells, which are hydronium, carbon dioxide, and 2,3-BPG. It turns out that all of these things favor the movement of hemoglobin from the R state to the T state. Okay? Why does that make sense? Well, if you think about why do we need oxygen? You've probably heard in your general biology classes that oxygen is the terminal electron acceptor in the respiratory chain, right? Meaning that if that respiratory chain doesn't have oxygen, your production of that significant number of ATP shuts down, right? So you're no longer producing that massive number of ATP. Instead, you're relying on 2 from glycolysis, which is not enough for a cell to live on, especially something that runs aerobically like the heart cells, right? So it would make sense that if you're actively metabolizing, you need more oxygen. So why not have byproducts of metabolism cause dissociation of oxygen and movement into the tissues, right? So if you actively are consuming oxygen, why not get more? So it turns out that these byproducts of metabolism, hydronium, carbon dioxide, and 2,3-BPG, get released by cells that are actively metabolizing. They bind to hemoglobin in the R state and cause it to move into the T state. So the oxygen dissociates, and just like concentration, the oxygen is going to move from an area of high partial pressure to low partial pressure. And what that means is that it's going to move from the blood into the cell. Okay? We're assuming that in the blood, if we're having a lot of dissociation, there's a high partial pressure of oxygen, so it's going to move to the cell where we're consuming it and where we're assuming that the partial pressure is lower. Okay? So these products bind to the protein hemoglobin and cause it to dissociate oxygen by moving it into the T state. Okay? Now, with H3O plus and carbon dioxide, we already have seen how this works. If we go back and we sort of review what we've seen, right? How did CO2 cause it? Well, it bound to the lysine residue, right? And you sort of have to view these proteins, although they're not enzymatic, you have to view them as dynamic proteins, dynamic structures. Anytime something binds to a protein, what happens? It changes conformation, right? Even something as minuscule as a proton, if it binds to a protein, guess what happens? It changes conformation, right? So if a protein binds to the histidine residue, right? If carbon dioxide binds to the lysine residue, what happens? It changes conformation, and that change in conformation facilitates the movement from the R to the T state. Now, that really doesn't answer our question about 2,3-BPG. Well, we come back here. Let's look at another picture. So if we have 2,3-BPG, or so we have hemoglobin, recall that it has 4 subunits, right? Okay. Now, if you look here in the center, there's this sort of hole between the globin chains, right? It turns out that 2,3-BPG is good at binding there. So 2,3-BPG will bind here in this hole between the globin chains, okay? And that change in conformation facilitates the movement of hemoglobin from the R to the T state. Therefore, oxygen dissociates and moves into nearby cells that are actively metabolizing. And really what you need to do when you're studying hemoglobin function is really try to reason through it instead of memorizing, right? If you have a cell that's actively metabolizing, what does it make a lot of? Hydronium and CO2. And one thing you'll get when you do glycolysis is 2,3-BPG. And if you've got an actively metabolizing cell, what does it need more of? Oxygen, right? So it makes sense that it moves from the R to the T state and causes dissociation of oxygen to the tissues, okay? Now, what we're going to start getting into now, and I'll do this in another video, is we're going to start getting into a concept called cooperativity. And I'll go ahead and define cooperativity to you now. And cooperativity is essentially when you have the binding of one ligand influences the binding of the next ligand. And we're going to go into excruciating detail in the next video on cooperativity. But suffice it to say for now that that hemoglobin exhibits cooperativity. And we mentioned in the myoglobin video that myoglobin does not. And part of the reason that hemoglobin exhibits this cooperativity is due to its tetrameric structure. Okay? But for now, the main functions I would say for hemoglobin that you, and really the red blood cell in general is number one, the heme binds oxygen. Okay? That is, that's something you should have learned in your biology classes, is that heme binds oxygen. Okay, that's the case in myoglobin and hemoglobin. Another thing you should understand is histidine residues on hemoglobin abstract protons. So if they abstract the protons, they raise the pH. And if the pH rises too much, then water abstracts the proton and you lower the pH even more. Okay? So they alter the pH by binding protons. Okay? So they act as a buffer. Okay? What else? Lysine residues bind carbon dioxide, right? And so you can transport carbon dioxide as a carbamate group on hemoglobin, okay? What else do they do? Well, they're responsible for transport of other molecules like nitric oxide, right? We didn't really go into a whole lot of detail on that, but just know they can bind other things. And also they protect, the heme protects against carbon monoxide poisoning. If you have lots of carbon monoxide in your blood, you don't want it getting into the mitochondria because it inhibits cytochrome C oxidase of the respiratory chain. So it binds carbon monoxide and prevents it from getting into cells. Okay, so it's a buffer, sort of, you could think of for carbon monoxide. Okay, and then with the red blood cell in general, we go back to our equation, which was what? It was over here, it was carbonic anhydrase. Now, this is not catalyzed by hemoglobin, but it is certainly associated with it. So when you think of hemoglobin and red blood cells, in your mind, think Le Chatelier's principle. And when you answer questions on an exam, definitely answer in terms of Le Chatelier's principle. It's going to tell your teacher that you know what you're talking about, but also it's going to help you rationalize why things happen in red blood cells and hemoglobin in general. So in the next video, what we'll do is we'll go in more detail with T and R state, and we'll talk about cooperativity in hemoglobin. I hope this video helps. See you in the next video.